Good morning, good afternoon, good evening, everyone. It's 10 a.m. EST from Clemson, South Carolina. My name is Dil Tavaraja. I am a professor here at the Clemson University. I oversee the Pulse Quality and Nutritional Breeding Program at the Plant and Environmental Sciences, Clemson University. Our program is funded by the National Institute of Food and Agriculture, ORAI program. To support this Going Organic platform, we breed organic pulse crops. Today is our second webinar for the 2025 Fall Going Organic webinar series. This webinar series highlights the organic production systems linking to phenomics, to genomics, and resistance breeding. We invite speakers worldwide to share their experience in plant breeding, genetics, management, nutrition, marketing, processing, and policy. So we are very fortunate to have these outstanding global speakers to coming into our webinar series and share their experience. Before introducing the speakers, just a little bit of housekeeping notes. Please introduce yourself via chat and ask all the questions via Q&A. So it is with my great pleasure to introduce today's speaker, Dr. Valerio Hoyos-Villegas. He's one of the rising stars in the U.S. and he's actually was the former assistant professor and the bean breeder at the Michigan State, not the Michigan State, in Canada. So now he's at the Michigan State. So he's an assistant professor of plant breeding and genetics in the Department of Plant, Soil, and Microbial Science at the Michigan State. And he leads the very famous bean breeding program from MSU, which is dedicated to developing high throughput performing or the phenomics to genomics and resilience to climate resistance and also nutritional quality, some market classes. He does quite a bit of quantitative genetics. He's one of the rising stars in the US for the quantitative genetics and the bean breeding. He has more than 50 peer-reviewed publications, many invited talks, as well as nationally and internationally recognized breeder. And also he's the past president for the NAPPN, that is the North American Plant Phenotyping Network. And I have been communicating with Dr. Valerio for a long time, and he has been tremendous help on the establishing our breeding platform for US back to bean breeding and keeping up with the industry. So he won the Early Career Award in 2024 for his outstanding research. So with that, I would really like to welcome you, Dr. Valerio. It's your floor.
Thank you very much, Dil, for the kind introduction and for the invitation to speak in the Going Organic series. It's my pleasure, and I'm very pleased to be here with you all. Yes, so as Dil mentioned, my name is Valerio Hoyos-Villegas. I am an assistant professor here at Michigan State University, but I'm also an adjunct professor at McGill University, which was my previous institution. So today I wanted to share a little bit of our insight and knowledge in our work in simulating simulating how to optimize genetic gain in common bean. So I changed the title a little bit because it's not just about genomic prediction, but rather about how to optimize genetic gain. And this is the work over a number of years now. So of course, I hope that I don't have to highlight this to our audience, but I feel it's always important to indicate what the challenge is, and that is that basically our yield trends are insufficient to meet global food production relative to population growth. So in order to meet this challenge, it's important that us plant breeders um, sort of meet the needs or the challenge um, by accounting, because we account for 50% of the increase in crop yields, as has been evidenced in previous studies. However, most of the characters that improve yield, that are controlled by major genes, I would say, have been in at least in many crops, but particularly in common bean, they have been highly leveraged, right? And the genes that control for productivity, such as yield, have yet to be maximized in their contributions. And this is a challenge because they are quantitatively inherited, and managing this type of variation in breeding programs is uh, uh, orders of magnitude more difficult. So how are we to increase genetic gain? How are we to speed it up to keep up with with the demand? So I'd like to start with a framework that oversimplifies, I would say, the breeding cycle, but it serves as a framework. And I would say that it can be summarized as three words: informate, evaluate, select, and repeat. Okay. And this kind of framework can be theorized by the response to selection equation, which is made up of gain over time, selection intensity, selection accuracy, which is a correlation between breeding value and phenotypic or true breeding value or phenotypic value. Useful additive phenotypic variation or standard deviation, and the time it takes to complete a breeding cycle. In practical terms, the increases in each one of these components can be implemented in things like F2 populations, screening nurseries, increasing the number of populations, reducing noise by improving their ability to evaluate in the field, managing ways of bringing in new variation into populations, which crosses new variants, new gene variants, etc., and shortening the amount of time between generations or cycles with shuttle breeding, marker-assisted selection, or genomics, and now genomic selection, depending on how it's implemented. So I always say that a breeding program is also a resource management exercise, and the breeder must make decisions that maximize the use of that maximize the resource efficiency use. Okay. And so, how do we implement these changes in order to achieve this goal? So, some of the things that we know are that plant breeding is a complex and long-term endeavor. It is logistically challenging to implement changes that cause dramatic shifts in the operations of a breeding program, and that can be costly and difficult to ascertain because because their effects are realized in the long term. So it's costly or even impossible to take this kind of risk and iterate failures and successes empirically as you would in an experimental setting. And of course, there are random effects of unknown unknowns in breeding programs. So as you try to implement these changes, there are other factors that are perhaps uncontrolled that we can't account for. So given the resources available to a breeding program, there are a number of questions that one might pose along the way in order to determine how to best or how to get to a better variety or a faster to a better variety. And this can be how many parents can I use? What's the selection approach? What's the breeding strategy? And do these any kind of changes result in long-term bottlenecks, etc.? And these are questions that we can then pose through computational means and simulation. So in this seminar, I'd like to present four what I call parameter-based questions. The first one is there a way to optimize overall genetic gain? Second one is, how can potential optimizations to genetic gain be implemented, provided that we find some results, some positive results in the first question, leads us to the second question. And now the third question, if these two are true, can optimized models be used to shorten breeding cycles? And finally, can these optimized models be used to extend to unseen environments? So the first question, is there a way to optimize genetic gain overall in common bean? And this is the work from my former student, Jennifer Lin, and our collaborators in Australia, New Zealand, North Dakota, and the University of Florida. The objective of this study was essentially to establish a set of baseline simulations and selection using various known selection strategies available to bean breeders, and identify whether there were differences in the performance of genetic gain. And this will enable us to, if that's successful, we can determine if changes in some of the compositions of these breeding programs, such as parental size, population size, heritability, have an influence on fixed favorable alleles, and whether some frameworks imposed above these selection strategies, such as genomic selection and speed breeding, can surpass conventional breeding frameworks as a comparison. The breeding strategies, we used 5 breeding strategies, and I won't go into the details of each one of these, but generally speaking, I'd like to categorize them into 2 major types of strategies. One of them is population-based, such as mass selection and bulk breeding, and other 3 are line-based approaches, such as single seed design pedigree method and modified pedigree method. Within our framework, we're able to establish a difference between the type of environment being grown based on error variances inputted into the baseline data. And these would be nurseries and replicated trials. And each one of these procedures that you see here would be one cycle, then is then repeated. As a reminder for those of you in the audience that are not familiar with genomic prediction is a procedure where we have genotypes and phenotypes that serve as a training set to train a prediction model that is cross-validated and tuned to resemble the characteristics of the training set. And then we have an independent test set that is applied, and then new genomic estimated breeding values are applied to conduct selections. So the potential advantages of genomic prediction are therefore that we would capture more of the portion of the genome that is highly heritable, and we reduce noise, which presumably would be the case with making selections directly for phenotypes. And we would also enable us to expand the capacity of a trial without necessarily having to increase area because of this first point of reducing the noise. We are testing only the materials that are most most contributing to accumulating useful additive variation. So if we apply it early on, we can also make earlier selections, and we can make more accurate selections. So with that out of the way, I'd like to then point out the data pipeline. I won't go into each one of the steps here of the information that we capture along the way, but essentially we wanted to start with a population that had some level of linkage disequilibrium established. So we generated a large base population that was randomly intermated through I don't recall at the moment, but it was in the order of hundreds of generations to generate some linkage blocks, some uniform linkage blocks. And this population was then used as the baseline sampling population in QGene. QGene enables us to input a set of information that basically dictates the control and the degree of complexity of genetic architecture, and this gives us a baseline population. That baseline population can then be subject to the procedures that we set at the beginning, And in order to set up the various frameworks, which would be conventional genomic selection and speed breeding, we would then subject that population to either the procedure or the procedure, but with genomic estimated breeding values, and then conventionally selected based on the breeding values and not the phenotypic breeding values, as you would in conventional. So based on GBBs and not TBBs, and then speed breeding, we simply just increase the rate of a generation time by about a generation and a half more per year. Okay. This gives us sort of three outputs. First one is the fitness of the population, the rate of fixation, and then the distance from the rate of fixation of favorable alleles and kind of an complementary output from fixation, which is the relative distance from the optimal individual with all favorable fixed alleles. So this is the overall simulation experiment. We have our 5 breeding strategies. We have 3 traits at high, medium, and low heritability with various QTLs collected from the literature that were highly representative. We set 4 initial parental population sizes, and then the 3 frameworks. So all in all, this was a total of 180 scenarios, and with replication and 50 replications and 10 independent runs averaged together, that was a total of 90,000 simulations. And we had the ability to do this because of our access to Compute Canada. In order to place the relevant QTLs, we use a consensus linkage map available in Beans. And so then with this we just initiated the simulation. So the results on the left here, we have the 3 breeding strategy, the 3 frameworks, and the 4 initial parental populations, and then each curve represents each breeding strategy. And this has genetic gain across 10 cycles. Here on the top, you can see the cumulative gain, which is what I'll focus on at the moment. And then on the right, we have the number of cycles until 95%, or basically most of the attainable cumulative genetic gain has been reached. So that gives you an average of the amount of time, relative amount of time necessary in order to exhaust variation in the breeding program. And here we have the average genetic gain, which is the total genetic gain divided by the number of cycles required to reach 95% of possible genetic gain. So in summary, the most effective overall procedure was speed breeding. It accumulated most of the variation in the fastest amount of time as one would expect, and this was particularly effective in the line-based methods such as single seed descent, pedigree method, and modified pedigree method. So now, to the breeders in the audience, that might not be a surprise. If you can select top alleles in a faster way, then of course you're going to accumulate genetic gain faster. The downside to this, I'll talk a little bit about it in a moment, but when we compare it with the other frameworks relative to the parental sizes, the rate of genetic gain with genomic selection at low parental populations was lower in genomic selection than in conventional. In other words, total final genetic gain in genomic selection was lower if the genetic base was smaller. If you increase the genetic base, that was similar. It's highly comparable. When we look at genetic gain or percentage of fixed favorable alleles, rather, between conventional and genomic selection, there was a big difference in the accumulation of favorable alleles in bulk or population improved methods, rather, in one versus the other. So one was much effective at accumulating favorable alleles than the other. So if we look at a mid-heritability trait such as white mold tolerance, genomic selection showed similar cumulative gains to conventional. And if we look at fixation of favorable alleles, single seed descent under speed breeding, it fixed 98% of the alleles for yield and 93% for flowering, while pedigree method generally showed the fastest allele fixation rate, and mass selection consistently resulted in the largest Hamming distance. So one major conclusion is that the optimal breeding scenario varied depending on the heritability, and in a relative change in delta G, or genetic gain per cycle, went up to 8.69% was achievable under optimal conditions. That was high initial appearance, high accumulation of favorable alleles, and low rate of loss of genetic variance. And this brings me to loss of genetic variance. If we look at how it changed over time with the three frameworks, a lower number of parents, variance decreased at a slower pace because of the lower frequency of available selection candidates. So therefore, this has a consequence on genetic gain, which makes it slower. At high initial parents, there's a higher frequency of selection candidates. So therefore, the distillation of favorable genetic variance is a lot faster with highly effective methods such as speed breeding and genomic selection, but the loss is far greater, as we can see here. So genetic variance generally decreased of the cycles simulated, but it happened at a much faster pace with the speed breeding and genomic selection, particularly in line-based methods. Okay. In comparison, bulk breeding maintained most of the genetic variance, and this is, even though this was maintained, that is also reflected in slower rates of genetic gain. In terms of genomic selection accuracy without model updating, the mass selection resulted in the highest accuracy, while it maintained higher accuracy and caused by slow favorable accumulation, meaning low population differentiation from the training set. So the study conclusions are that in this first study, that small breeding programs, meaning low initial parents, may not benefit from genomic selection in dry beans. So if you have a narrow genetic base, you'd have to look at it very carefully. Single seed descent was most effective for days to flowering, where single seed descent pedigree or modified pedigree were observed resulted in the greatest benefit when you have high heritability or intermediate heritability. And conventional and speed breeding generally led to greater cumulative genetic gains. Genomic selection performed similarly conventional breeding. In some cases, genomic selection resulted in more genetic variance being maintained, very few exceptions. And this means that the complexity and success of breeding programs when implementing these methods depends on the strategy, framework, and characteristics of the breeding program. And speed breeding was the most efficient methods, but raises concerns with loss of genetic variance in the short term. So now moving on to the second parameter-based question: how can we then take these lessons from optimization of genetic gain, and how can we implement them in a breeding program? This is the work again from Jennifer and my other student Isabella Chiarabalotti and our collaborators. This is a rather smaller experiment with very simple question, which was how, based on these baseline strategies, can we implement genomic selection model updating, and does this have an influence on genetic gain and prediction accuracy, and to what extent is this a durable approach? So we took the same set of simulations where we have phenotypic. We can simulate true breeding values. We simulate genomic estimated breeding values. Again, when we run through our BLUP, we basically conduct selection based on the same framework, but the input information are the genotypic estimated breeding values. So we would then update the model based on a sample of selected individuals from the third cycle to estimate new marker effects. Okay. So we had two intended accurate estimates of genomic selection accuracy. One which we called in silico realized genomic selection, which is simply a correlation between the true breeding value and the genomic estimated breeding value, and then an expected accuracy, which has been reported in the literature. based on this equation. And the idea is that we can predict what the accuracy will be in following generations. But because we have information from generation to generation, we can determine the whether that expected accuracy correlated with the observed accuracy. Okay. Um, so the first hypothesis, genomic selection strategy influences genomic selection accuracy and genetic gain. So realized accuracies, or called in silico realized. So from basically the direct correlation between GBBs and TBBs fluctuated depending on the trait, genetic architecture, breeding strategy, and the number of initial parents. And trait architecture and breeding strategy had a greater impact on accuracy. And initial parents also had an effect on accuracy primarily among population-based methods, as you can see here in mass selection. There's a big shift as you increase number of parents. There's a big shift in accuracy in mass selection. High heritability also had and high initial parents, so therefore resulted in the most durable accuracy. We are not able to obtain consistent expected accuracy calculations from expected accuracy formulas. Now, the hypothesis 2, genomic selection model updating influences genetic gain and prediction accuracy to varying degrees depending on selection strategy, trait heritability, and number of initial parents in the breeding program. So the impact here was that model updating generally improved prediction accuracy, therefore benefiting population selection methods more than line-based methods. So in general, even though model updating did lead to a sharp increase in genetic gain, the distance was the genetic distance from the training population was already so large that it followed a sharp decline, obviously depending on heritability. So there are factors that influence accuracy, as we know, like genetic gain, like yield. In the case of yield, so the genetic complexity influences accuracy greatly. But this plateau, the plateauing and change in genetic gain is likely due to the faster fixation of alleles and diminishing genetic variance, causing more genetic distance from the training population at a faster pace. So there's a complex interplay here, which makes it difficult to develop durable genomic selection accuracies and durable and sustainable rates of genetic gain. We're talking about long scales here, right? In the scale of multiple cycles of breeding. Okay. Effective implementation requires careful consideration of training population size, marker density, and the necessity to regularly conduct model updates. And mass selection and bulk breeding were the best methods for maintaining genomic selection accuracy, while line-based methods were more compatible with genomic selection for maximizing genetic gain, but at the cost of a far faster loss in accuracy. So there is no one size fits all solution for genomic selection, and its implementation requires careful attention. So this, let's say this idea that we are in the search of a highly durable, robust, generalizable genomic selection implementation accuracy, and led us to this idea of implementation pathways. And this is the work of Isabella. It was just accepted in the Plant Genome. Can we then optimize these models to shorten breeding cycle and maximize genetic gain? So the objectives here were to optimize genetic gain for yield while maintaining genetic diversity. As you see, this is sort of one of the major lessons from the previous two studies, are that yes, we can maximize accuracy, or we can maximize genetic gain at great costs in loss of genetic diversity. So can we maintain both? And we wanted to also explore the practical implementations of genomic selection in a breeding program by simulating different pathways, focusing on, for example, when to train a prediction model, when to select parents for the following cycle, which generation to collect training data on, and whether linear regression or a non-parametric model for estimating breeding values was more effective. In this case, we switched to using AlphaSuite um, from Gregor Gorjan's group at the Roswell Institute. And this here we wanted to we utilized 4 QTLs, evenly spaced QTLs on each one of the 11 chromosomes. So we made it a little bit more complex, and this led us to an estimate of narrow sense heritability for yield, about 0.14. We compared neural networks and ridge regression, or RBLUP, and 3 different training sets. So one with F2 data, one with F5 data, and a mix of F2 and F5 data. So those are the 4 pathways. First pathway was we then generate parents. So the overall, every pathway went through the same procedure. We generate parents, and then we self to F2 derived F5 family selections. That goes into preliminary yield trials and then advanced yield trials. Pathway number 1 would then take F2 data, so train at F2 and select parents at F2, and then generate a new cycle by doing rapid cycling and selecting parents, and then going forward. Pathway number 2 would then generate a new cycle at F2, but it would use with training data at F2 derived F5, but select parents at F2. So continue rapid cycling, but use information from more elite advanced lines from more advanced generations. Pathway number 3 would then train at F2, but then select parents at F5. And pathway number 4 would then train at F5 and select parents at F5. Okay. For the results, if here we have breeding cycle on the x-axis, and then percent change in genetic value, or change in genotypic and genetic gain, essentially, between control, no change, and then the different pathways, okay? So here, for example, if we look at pathway number 4, so that was, if you remember, pathway number 4 is select and train at F5. There was very low resemblance to test, so therefore the genetic gain was very low. Whereas if we switch to F5, pathway number 4 with F5 data, there was a much higher resemblance to test. Okay, so pathway number F2 with F2 data with pathway number 4 was low resemblance to test, and pathway number 4 with F5 data obviously had high resemblance to test. So that is kind of not surprising. And when we look at the mixed data, there was mixed resemblance to test. So then you get kind of an average of the two. So that is, it tells us that the simulations are kind of working as we would expect. But more interestingly, neural networks resulted in consistent genetic gain regardless of data resemblance. Okay, so this indicates a greater level of learning from the data and model robustness. So this supports that, of course, that the training data should resemble the test data as much as possible. Now, if we look at average genetic gain per cycle, early generation parent selection, such as rapid cycle selection, generally led to higher gains. And then the 2 F5 models were highest when implemented obviously at F5 and used to select F5 data. If we look at here, the highest genetic gain was obtained with ridge regression and with mixed data, while F2 data was used for training, pathway 1 with F2 data resulted in 16% gains, and pathway 4 with F5 data resulted in 11% average gains. It was quite high. So ridge regression generally always yielded the highest gains, while neural networks showed the more consistent gains over time. In other words, ridge regression was very effective at maximizing gain, but perhaps it was very specific. It was very specific to the type of data that it used as input, while neural networks allowed for perhaps at the cost of a slightly lower gains, it allowed for more generalizable use of the model. In terms of accuracy, our blob showed more consistent genetic GBV prediction, while non-parametric models fluctuated, resulted in both highest and lowest prediction accuracy. In genomic prediction models, training data therefore needs to be as diverse as possible to match the test. One interesting finding here is that the total change in genetic variance fixed in the case of neural networks at F2, okay, fixed about over 90% of the favorable alleles possible. So showing that that kind of goes back to that same idea that it was capable of developing neural nets much more effectively and generalizing across the model to capture most of the favorable alleles. So non-parametric models demonstrated a greater balance of maintaining allelic diversity and genetic gains, indicating the potential for use. However, there was a great loss of genetic variance when the model basically maximized its ability to conduct this to conduct selection. Conclusions: genetic selection pipeline maximizes benefits when a diverse training set is used in linear regression, while parametric models, regularized parametric models, outperform non-parametric models. But in some cases, neural networks can have superior performance and better maintenance of genetic variation. So this indicates that frequent crosses, generating new recombinants, and renewal of genetic variants is encouraged by these approaches. Okay, so one of them allows for less loss of depleting this variation, while the other one maximizes it. And then some of the next questions are: can we then stabilize and develop high-performing non-parametric models for optimizing genomic selection pipelines? And while genomic selection holds significant promise for increasing genetic gain, its implementation still presents some challenges, I think. The final question is: can optimized models be used then to extend this existing information to unseen environments? In other words, can we make better use of the resources available to optimize selection for other environments? Okay, so can we take then pooling multi-location data, help bypass the data volume bottleneck to increase prediction and increase the generalization of both of particularly non-parametric models, but also maintaining the durability of linear models? As we've seen, they have significant loss very quickly. So the objective here was to see if utilizing ensemble models for genomic prediction was effective. Can we identify how model accuracy varies across locations, and do these ensemble models are useful in the estimation of the genotypic value of previously observed genotypes in new unobserved environments? And finally, we wanted to understand what was the relationship between environments that then causes this variability in accuracies in unseen environments. So we had three primary models: linear regression, ridge regression, and neural nets. Okay. I won't go through the description of these, but essentially we had the three models that we wanted to test: parametric versus non-parametric models. And we had three compositions of the data sets. Essentially, we had I think I go through these individually, but so just so you get an idea that these were sort of the major frameworks. And then we had a multi-environment training data set, which is the Common Dry Bean Nursery, which is a nursery that has been shared across the U.S. as a public data set that spans 70 locations over 30 years with about 150 phenotypes, hundreds of genotypes, and it contains 1.2 million SNP markers. Okay, so the singular model, we have three types of ensembles. The first one was a singular model. So basically, we have a training set with multiple locations. They all go into a singular model prediction, and then that is used to infer onto a test set. So there is no discrimination, there is no selection of locations. They all just, it's the average of all these, each one of those models. And then we compare true and estimated value of with just Pearson correlations. Then there's the ensemble model. We then take each location results in a single model, and then the average prediction of all the single models, which is the ensemble, then is applied onto the test set, and then again we check for accuracy. So the ensemble then averages the predictions from all these submodels to produce the final output for each observation. And then finally, we tested an optimized ensemble test, which is essentially we run the single models, but then cross-validate and determine which one was the an optimized set with the highest accuracy, and then those become an average prediction, and then applied onto a test set and compare. We estimated heritabilities across all these environments, and we found that seed yield was low and seed weight was high, so we kind of used those two for contrast on average. And so results here on the top left, we have seed weight with linear regression and seed yield with linear regression. As you can see from very obviously, the prediction accuracies were highly contrasting. And you can draw a very simple conclusion, which was that as you have differences in heritability, then therefore there is no difference whether you use ensemble versus not versus linear single model. So there's almost no difference. However, the best models for seed yield, in the case of seed yield and implementation of bridge regression, were the best models. Okay. So prediction accuracy mostly comparable, better performance in high heritability, and in some environments heritability was lower than prediction accuracy, meaning that genomic selection was more accurate than phenotypic selection, but that was depending on the environment, and we'll get into that a bit later. Now, if we look at linear regression, prediction accuracy of linear regression with the ensembles, okay, so there's a major advantage to prediction accuracy under high heritability and low genotypic variance. If we compare this prediction accuracy with the overall phenotypic variance of these within each environment and across environments, there's a big advantage when you have high heritability and low overall environmental genetic variance. In contrast, there's an environment-specific advantage to prediction accuracy under low heritability, such as yield, and high environmental or total genetic variance. Okay. And so what we concluded was that if we can separate environments based on their relative the relative ability to discriminate between genotypes. In other words, their imposition of for whatever reason, their environmental characteristics led to a greater imposition of discrimination among genotypes, such that it maximized variance in the trials. Highly discriminated environments, or the ones with high variance, resulted in high singular model accuracy, whereas less discriminated environments, those with lower variance, showed high ensemble model accuracy. So there's an advantage there. If you have more stable environments, there's a high ensemble. There's an advantage there. So, for example, here, if we look at say Kansas and Arizona. So Arizona had very high phenotypic variance, so that which made it a very high discriminating environment, and therefore the optimized ensemble and singular model had minimal advantage. The prediction accuracies were low, and same thing with Kansas here, for example. But if you look at, in contrast, environments with low phenotypic variance, such as Manitoba here, that exceeded its variance was exceeded by prediction accuracy, therefore showing a potential advantage there. And this is what I'm showing here in the case of, for example, North Dakota and Manitoba. So there's a bias variance trade-off. So some ensembles mitigate this bias variance trade-off caused by discriminativeness, and they allow for a tighter model fit without introducing high bias, especially in those environments with low phenotypic variance. And it's we attribute this to the ensemble's ability to reduce overall model variance by averaging across submodels. So neural nets, when using neural networks, the ensemble approach is often outperformed the singular model. Highly heritable traits tended to correlate with higher prediction accuracy, which is, I guess, good news for selection for high heritability. And pooled data from this multi-environment trial yielded high prediction accuracies up to 0.7 for flower and up to 0.67 in some locations, again, based on this bias variance trade-off. And this highlights that if breeders collaborate in pooling data, this can overcome the bottleneck of low data volumes for training data sets, particularly for small breeding programs. And so this demonstrates that environment ensemble models, particularly optimized ones, can be effective for genomic prediction in common beans, especially in those highly stable locations. And collaboration, again, leads to a synergy in the implementation of genomic selection. The overall conclusions from the study are that there's a need for continued optimization of plant breeding pipelines in order to maximize genetic gain. There is simulations serve, of course, as a way of estimating it and assessing change without having to run the experiments and allow us to gain a baseline and decide. But they are a strategic tool. They are not a tactical tool. They're more a high-level kind of approach. And simulations, they provide us with a set of outputs, and they allow us to make, therefore, decisions based on the upper ratio of successes versus failures across averages of multiple or hundreds of runs. If we continue to try and investigate and develop high-performing, high-accuracy, stable non-parametric, for example, something that we're looking in my lab right now is the use of transformers and pre-trained models that can learn more and more from larger data sets. This may enable us to further optimize genomic prediction accuracy and durability in pipelines to ensure accurate identification of top-performing individuals. But ultimately, this is the quality of the information that goes into these models dictates the quality of the predictions, of course. And this can be an avenue, one avenue that can be utilized to optimize this could be the use of phenomic data. I'd like to acknowledge our two labs, our lab here at Michigan State and our lab at our sister lab at McGill University, and our funders both in the US and Canada. Thank you very much.
Thank you so much, Valerio. It was wonderful and it was very informative. There are a couple of questions in the Q&A, but before we go to the Q&A, I wanted to ask you a question because very interesting about how you simulate all these models and you link the genomic predictions into the different heritability aspects. So like it's based on the traits. So what is your advice for the new breeding programs or the new assistant professors who start in breeding programs? So do you have any advice for them? Because I see some.
Yeah, I say, yeah, well, I went through that experience myself, and this is what led me to all these questions. So I established a breeding program in Canada from scratch, uh, and there's some lessons learned there. Some of them, of course, the first obvious one is try to start as broadly as possible in terms of germplasm. Try to introduce as much elite diversity as possible. Of course, this is easier said than done, but as you can see for example, from our simulations, there is one of the greatest advantages dictated by the extent of the genetic base, right? So if you can establish a wide genetic base of elite variation in the beginning, that has huge positive long-term consequences. the second piece of information is, of course, I said, breeding programs are also a resource management exercise. So it can be easy to get bogged down in the numbers. There is, even though I try to put things in a very quantitative framework here in this talk, there is an element of a sense that is developed by the breeder in terms of managing genetic variation, which introduces um, uh, some would argue a, a big part of the element of success is understanding the inherent variation, and this is what I mean by unknown unknowns, is that when we make crosses, we don't always get the expected variation that we aim for, and this can have an impact in the overall in the patterns or the changes that we see from cycle to cycle or generation to generation, and this is something that needs to be kind of managed and understood by the breeder. Um,
That's
and
right.
have a very good relationship with your industry to establish your goals. Yeah, that would be the last one.
Yeah, yeah, that is actually wonderful. So let's go to the Q&A and then we'll come back to the other questions as well. So this is from our serial breeder. I don't know whether you can see it. It's the genomic prediction models typically demonstrate a small difference among them with respect to prediction accuracy. Can you comment on how important a small increase in prediction accuracy is within context of the forward breeding program? I think he mean by the
Right.
cereals, but
A small increase in prediction accuracy is okay. So like, let's if we have two models and there is that one has say 0.6 and the other one has say 0.7, does that make a difference? I guess if I understand the question correctly. Yeah. So I would say generally no. But here is I guess the way I would explain that is here. So here we have the mean heritability. This is the environment heritability, the average heritability, okay? And here we have the individual location heritability, okay? The way I would rationalize this is heritability is an indirect selection accuracy for phenotypic selection, right? So if you have high heritability, that means that your ability to select based on phenotype is presumably high because you're capturing more of the additive effects more effectively, okay? And prediction accuracy is a similar statistic, okay? Now it's not the same statistic, and they're hard to compare, but it's as close as we can get to comparing them, okay? So that difference that I would indicate would be on the basis of that kind of comparison, if there's an advantage, okay? So if I have a model that can surpass the heritability for the trait on average across my testing environments, then there is a case to be made there for the implementation of genomic selection. In other words, conversely, in cases where heritability, say for example here, where heritability is far greater than the accuracies I'm able to obtain, then perhaps that would be counterproductive.
right. That would be fantastic. And there is one more question. We are running out of the time. So does applying rigid regression, that is
Well,
penalizing
yeah.
the coefficient closer to zero, would add unintentional bias to the output?
Yeah, this is the effect those effect we're actually looking at that in this transformers experiment that we're running right now. So I would say that the applying or fine-tuning coefficients does have a great effect, but that effect is much smaller than the relative effect of training data sets. Okay, so you can
Yes.
fine-tune them, but that the relative change in the outcome of genomic prediction accuracy will not be as great as the relative change in better, or rather more tightly correlated uh, marker and phenotypic data that goes into the model, if that makes sense.
Yes, that makes sense. Thank you so much. And I think we learned a lot about your exciting modeling, and it is wonderful to see how you can apply these things into our crop, because legumes are usually orphan crops, has not much of the genetic value, and then not much research done. But with your excellent research program, we can go forward. So this is wonderful. So next, and thank you so much, Dr. Valerio, for joining with us. And next, our seminar is on November 12th after our national meeting. And we are also bringing another rising star, just like Dr. Valerio, Dr. Nonoy Bandalio. He used to be our pulse breeder. He will be now working on cereals, and he will share more on the predictions and how we can go forward with the breeding. So thank you so much again for joining, and you all can go to our YouTube channel, and Tristan has linked into that. It's a Clemson Pulse Breeding YouTube channel, and whoever registered can watch this website locally. And thank you so much, and we will see you again in November. Goodbye.